Hello everyone, it's Eric from Stanford University and Strong Medicine. Today I'm talking blood types. By the end of this video, you'll be able to describe the biology and genetics of blood groups, specifically the ABO and Rh systems, and to describe why we need to worry about blood groups and blood typing at all, specifically focusing on hemolytic transfusion reactions. Before jumping into the biology though, let's start with some history. Prior to the discovery of blood types, or any knowledge of the immune system, some doctors performed some treatments which to us in modern times might seem kind of outlandish. When transfusions were initially attempted in the 17th through 19th centuries, they typically involved directly connecting the donor and recipient without any consideration for blood compatibility. In fact, in the very earliest days of transfusions, some experiments involved cross-species transfusion, including the introduction of animal blood into people. You might guess how that worked out. Blood transfusions were frequently fatal and were seen as fringe medicine. That is, until Austrian physician Karl Landsteiner discovered the ABO blood group system in 1901, for which he later won the Nobel Prize in 1930. He initially discovered 3 blood types, which he designated A, B, and C. C was later renamed O, which is said to have stood for ohne, the German word for without. After this discovery, there was an explosion of interest into blood types, and transfusion medicine as a field was born. The first large-scale use of blood transfusion was done during World War I. Since untreated blood coagulates extremely quickly upon removal from the body, donors and recipients still initially needed to be in the same room, which was logistically challenging on the battlefield. Luckily, the introduction of sodium citrate as an anticoagulant during the war allowed for blood to be safely stored for hours to several days, which led to improvements in battlefield mortality rates. So, what exactly are blood types? Why was it, in retrospect, a terrible idea for doctors to directly connect the veins of 2 unmatched individuals? Well, it has to do with what's happening on the surface of red blood cells. If we zoom way in, we'll find that the surface is coated with various proteins and carbohydrates floating within and on the outside of the phospholipid bilayer that's the foundation of the cell membrane. These proteins and carbohydrates act as antigens, which can trigger an immune reaction if the body's immune system identifies them as foreign. This isn't a problem for our own blood cells, but when another person's blood cells are introduced into our body, if the antigens on those cells are different than the ones which our body's immune system recognizes as belonging to us, that's a problem. I'll come back to the immune response in a few minutes. A blood group system is a collection of one or more antigens that are controlled by either a single gene or a cluster of closely linked genes. For example, the ABO system that Dr. Landsteiner discovered, as well as another important system called the Rh system. These are just 2 of the 35 recognized groups, though most of those are of minimal routine clinical relevance. In distinction from a blood group, a blood type is the combination of clinically relevant antigens on the surface of a person's red blood cells that determine their compatibility during a blood transfusion. We can think of a blood type as an RBC phenotype. Examples of blood types include O positive, A positive, AB negative, etc. Now, let's take a closer look at the ABO system. This system involves carbohydrate antigens attached to a variety of transmembrane proteins. Enzymes, called glycosyltransferases, are responsible for creating the so-called H antigen, also known as the O antigen, consisting of 4 sugars in this specific order: galactose, N-acetylglucosamine, galactose again, and a sugar called fucose, not fructose, but fucose. The single ABO gene locus encodes an enzyme that catalyzes the final step in the synthesis of the A and B antigens. Which antigen or antigens are formed depends on which version or versions of the ABO gene an individual carries. The A version results in a transferase that adds N-acetylgalactosamine to the H antigen, giving us the A antigen, while the B version of the gene results in a transferase that adds another galactose. The A and B versions of the transferase differ by just 4 amino acids. And the O version of the ABO gene encodes an inactive transferase that leaves the H antigen unmodified. It seems highly likely that these carbohydrate modifications serve some biological purpose, but that purpose is, as of yet, unidentified. Let's take a closer look at the genetics. The inheritance pattern of the ABO blood group exhibits something called codominance. We know that there are 3 versions of the gene, also known as alleles. A and B are the dominant alleles, and O is the recessive allele. This results in 6 possible combinations of the 2 alleles, which are called genotypes: AA, AO, BB, BO, AB, and OO. These genotypes reflect the fact that every person has 2 versions of the ABO gene in each cell, one inherited from each parent. So what happens if both versions of the gene or alleles are A? In other words, the AA genotype. Well, the person is producing the A version of the transferase, so A antigen gets made. That's what's called the phenotype or blood type. What antigens are found on the surface? So this person would be blood group A. But what if their genotype is AO, one version of the active A transferase and one version of the inactive form? Since the active A transferase is there and working to make A antigen, the concurrent presence of the inactive form is kind of irrelevant. So individuals with the AO genotype also have the A phenotype. We can repeat this for BB and BO. For individuals with the AB genotype, they have versions of the A and B transferases simultaneously, so they get a mixture of the A and B antigens expressed, which gives us the AB phenotype. And last, if the individual has the OO genotype, they only have 2 inactive transferases present, so the H antigen never gets modified. Thus, these patients have neither A nor B antigens and are called phenotype or blood type O. Remember that the O stands for the German word for without. These patients were thought to be without ABO antigens, which we now know isn't exactly true since they still have the H antigen. The complexity of the ABO system results in a complex relationship between the blood types of parents and the possible blood types of their offspring. I'm not going to go through this whole chart, but let's look at a few examples. First, if both parents have O blood, that means they must have the OO genotype. Their offspring will inherit one O allele from each parent, and thus the child must necessarily also have the OO genotype and thus be type O themselves. Instead, if both parents are type A, their offspring could either have genotype AA, which would be blood type A, genotype AO, which would also be blood type A, or genotype OO, which would be blood type O. The last example I'll give here is if parent 1 is type A and parent 2 is type B. In this case, the offspring could have any genotypes but AA and BB, and therefore they could have any of the 4 blood types we're currently considering. Here's the rest of the chart. You can pause the video and work through it more if you'd like. Now let's move away from genetics to more discussion about why blood types are relevant. To review the 4 ABO blood types, we have type A, in which RBCs are coded with A antigen, type B, in which RBCs are coded with B antigen, AB, in which they have a mix of those 2 antigens, and O, which lacks both A and B. A person's immune system produces antibodies against the antigens which are not expressed on one's own cells. This means that individuals with type A blood are carrying anti-B antibodies, and those with type B blood are carrying anti-A antibodies. AB individuals carry neither, and type O individuals carry both. What happens when blood cells with one antigen are introduced into the circulation of a person carrying antibodies against that antigen, for example, via a blood transfusion? The person develops something called an acute hemolytic transfusion reaction. The transfusion recipient's circulating antibodies bind to the antigens on the donor's red blood cells. In some cases, this will activate the complement system, a collection of interacting proteins that, once triggered, release pro-inflammatory cytokines, stimulate a class of cells called phagocytes to remove cells that have been identified as foreign, and activate something insidiously called the membrane attack complex, which directly leads to cell lysis and death. The net result is hemolysis of the transfused cells, which means the cells are broken apart and their contents are literally emptied directly into the liquid, non-cellular part of the blood called plasma. This includes molecules of free hemoglobin. While hemoglobin within the red blood cells are essential for the delivery of oxygen to the body, that's why patients are being transfused in the first place, free hemoglobin let loose into the plasma in large quantities is a disaster. Hemolysis of transfused blood is far worse than if the recipient had never received a transfusion at all. The very first clinical consequence of this is usually fever and chills, which can be rapidly followed by hypotension, hemoglobinuria, meaning the presence of hemoglobin in the urine, which can lead to kidney failure, and a hematological complication called disseminated intravascular coagulation, which is widespread inappropriate activation of the blood clotting pathway. In other cases, the primary antibody involved in triggering the immunologic response is not able to activate the complement system, in which case hemolysis still occurs, but occurs more slowly within the liver and the spleen, rather than within the blood vessels. The speed of onset and overall severity of hemolytic transfusion reactions is dependent upon which red blood cell antigen in the donor is responsible for the incompatibility, the concentration of that antigen on the cell surface, the titer or amount of recipient antibodies already present that are against that particular antigen, the class and subclass of antibody, and whether those antibodies can bind to complement, which determines whether hemolysis occurs within the intravascular space in the blood vessels or in the extravascular space of the liver and the spleen. On one end of the spectrum of severity, acute hemolytic transfusion reactions can be a medical catastrophe carrying an overall 10% risk of death. Luckily, because of safeguards surrounding the blood transfusion process, this is an extremely rare event in the United States in the 21st century. The risk of death from an acute hemolytic transfusion reaction is about once in every 2 million units of blood transfused, which is more than 100 times lower than the risk of death from routine screening colonoscopy, and even a little lower than the risk of death from a routine tooth extraction. Now let's move on to discuss the other major blood group system, the Rh system. The Rh originally stood for rhesus factor. When the first antigen in the system was discovered, it was originally believed to be similar to one found on the red blood cells of rhesus monkeys. The Rh system is now known to include at least 50 different antigens on non-glycosylated transmembrane proteins, among which 5 of them, uppercase D, uppercase C, lowercase c, uppercase e, and lowercase e, are important. The D antigen is part of the RhD protein encoded by the RhD gene, while the C and E antigens are part of the RhCe protein encoded by the RhCe gene. As with the ABO antigens, the specific function of the RhD and RhCe proteins are also unknown, but based on their structure are believed to be ion channels. Now, as complicated as the ABO system might have seemed, that involved just 3 antigens. The Rh system is substantially more complicated. But luckily, while the mentioned 5 antigens are the ones that are at all clinically relevant, most of the clinical relevance of the Rh system is dependent upon just the presence or absence of the D antigen. Therefore, when a person is colloquially referred to as Rh positive, it means their RBCs express the RhD antigen, and when a person is referred to as Rh negative, it means their RBCs do not express the RhD antigen. The D antigen is inherited as a dominant trait. If a person expresses no Rh antigens at all, no D or C or E or any of the minor ones, they are referred to as Rh null, which is possibly the rarest blood phenotype in the world, with only about 40 individuals ever identified. As with the ABO system, the Rh system can also trigger hemolytic transfusion reactions, but in this case, it usually occurs after 24 hours and therefore is known as a delayed hemolytic transfusion reaction. The manifestations can be similar to the acute form, but usually not as severe and not as likely to be fatal. In addition to avoiding transfusion reactions, there is another huge reason that the Rh system is clinically relevant, and that is pregnancy. Individuals who are Rh negative, that is, lacking D antigen, do not typically produce anti-RhD antibodies until they've been exposed to D antigen either during a mismatched transfusion or more commonly in women during pregnancy with an Rh positive fetus. Maternal immunization against RhD can occur due to transplacental fetal-maternal blood exchange, which may happen either before or during labor and delivery. If an Rh negative woman now has anti-RhD antibodies, future pregnancies with an Rh positive fetus are at risk of leading to a condition called hemolytic disease of the newborn, in which the mother's antibodies attack the fetus's RBCs, leading to significant morbidity and occasionally fetal demise. To prevent this, all Rh negative pregnant women should be screened for anti-RhD antibodies at the first prenatal visit and again at 28 weeks. If antibodies are not already present, anti-D immunoglobulin, marketed in the US as RhoGAM, should be given at 28 weeks and shortly after delivery, unless the baby is known to be Rh negative. The point of giving anti-D immunoglobulin to Rh negative pregnant women is not to prevent hemolytic disease of the current fetus, but rather to prevent it from occurring during the woman's future pregnancies by preventing her immune system's response to an exposure to D antigen that might occur in late third trimester or during labor and delivery. So now that I've discussed the ABO and Rh systems, you should be able to predict the transfusion compatibility between blood donors and recipients. As with the ABO inheritance chart, I won't go through this whole chart either, but let's just look at a few examples. A donor who is O negative has neither A antigens, B antigens, or RhD antigens. So in the absence of any major RBC antigens, their blood is safe to transfuse into any of the other conventional blood types. Thus, individuals who are O negative are sometimes referred to as universal donors. In contrast, if we look at a recipient who is O negative, they'll be producing anti-A, anti-B, and anti-RhD antigens. As a consequence, they can only receive blood from an O negative donor. If the donor is AB positive, they have A antigens, B antigens, and RhD antigens, so they can only donate to other AB positive individuals. Whereas if the recipient is AB positive, they have neither anti-A, anti-B, nor anti-RhD antigens, so they can receive blood from anyone. Thus, individuals who are AB positive are referred to as universal recipients. And on your own, you can work out the rest of the chart. While there are 8 conventional blood types when the ABO and Rh systems are considered simultaneously, there are some unconventional blood types. I mentioned one previously, Rh null. I'm going to talk about one more of them, the Bombay phenotype. If we go back to the structure of the H antigen, also known again as the O antigen, before A or B transferases altered it, it looks like this, with 4 sugars in a chain. In the Bombay phenotype, a lack of an enzyme called fucosyltransferase results in the last sugar, fucose, not making it onto the chain. So affected individuals not only lack A and B antigens, but H antigens too. So these individuals produce anti-A, anti-B, and anti-H antibodies. This is a huge problem for them because red blood cells from all other people express some degree of H antigen, and thus, as with Rh null, people with the rare Bombay phenotype can only receive transfusions from other people with the same. How rare is it? Well, from its name, you might correctly guess that it's most common in India, where its prevalence is about 1 in 10,000. It's also relatively common in Pakistan, Iran, and Taiwan. However, among Caucasian individuals, its prevalence is approximately 1 in a million. If an individual known to have the Bombay phenotype is to undergo elective surgery, they can bank their own blood at the hospital ahead of time. But if it's an unplanned emergency, often the only source of blood is the emergent donation from a family member. There are even reports of Bombay phenotype blood being flown between countries when compatible family members could not be located, as also happens with Rh null. I'm now at the end, so let me summarize what we've learned. Blood types are phenotypes based on patterns of antigens present on the surface of red blood cells. The most clinically relevant antigens are the ABO and Rh blood group systems. The ABO system is composed of 3 alleles, A, B, and O, of which A and B are codominant, 6 genotypes, AA, AO, BB, BO, AB, and OO, and 4 phenotypes, A, B, AB, and O. The Rh system is composed of dozens of antigens, of which the presence or absence of Rhd antigen is the most important. And finally, the transfusion of mismatched blood can result in potentially fatal hemolytic transfusion reactions. Thanks for watching. Be sure to like and share the video if you found it helpful, and subscribe to Strong Medicine for more educational videos on medicine and health.